Well, thanks to the organizer. So I'll tell you about some work we've been doing developing statistical methods to perform epigenome-wide association studies. Okay, so as many of you here are very familiar with, so genome-wide association studies, or GWAS, has made quite substantial progress last many years in basically disentangling genetic variants, especially common genetic variants that are associated with human disease and phenotypes. And the statistical community has done a very good job in developing sort of rigorous tools both for designing GWAS studies and also for analyzing the results of GWAS studies. So, but in recent years, especially in the last few years, and also due to the work of many people in this room, we're starting to realize that there are many epigenetic contributions to many of the human phenotypes and diseases of interest, right? So that's beyond simply genetic variations. So there's becoming an increasingly need to try to understand these epigenetic variants across the human populations associated with disease. So the work that I'll describe here, so I want to describe some of the methods we've been developing to try to perform these epigenome-wide association studies, and also to lay out some of the challenges and opportunities for this direction of research. I mean, the goal is really to, hopefully we'd like to develop a rigorous set of statistical methods to perform epigenome-wide association studies that's analogous to what we have for the genome-wide association studies. So here's the outline of the talk. So I'll first just describe what exactly the epigenome-wide association is and outline some of the opportunities and challenges in terms of computational methods. And then I'll spend most of the talk describing a particular method that we developed recently called eWasher to perform some of these epigenome-wide association studies. And lastly, I'll talk about applications of these methods to DNA methylation studies and linking that to rheumatoid arthritis. So the goal of the epigenome-wide association study, or EWAS, is really to identify changes in the epigenome that's associated with disease. So this could be changes in terms of DNA accessibility, in terms of DNA methylation, or histone modifications, or a host of other things. Right, so the methods that we'll describe here will be applicable to general types of epigenomic signals. But for concreteness, I'll just focus on DNA methylation as a case study. And the idea is that we want to, given a specific marker of interest for epigenome, we want to scan across the human population, compare some disease phenotype, i.e. compare people who have some disease phenotype with healthy controls. And we typically assay the epigenome using either sequencing or from chip arrays. So conceptually, the EWAS setup is very straightforward. The idea is that we have a set of individuals with some disease and healthy individuals. So here we focus on the case where we have binary phenotype, but this will also be extended to continuous phenotype. And if we're interested in DNA methylation, we just assay the DNA methylation values across the genome, right, across many CPGs. So the genome here is basically corresponding to the x-axis, and each of these bars basically represents what is the DNA methylation value of particular CPGs across the genome. All right, the idea of the study is that once you've collected this large matrix of samples, you simply scan one by one across different CPGs and try to find which CPGs are differentially methylated between the cases and controls, right? So it's very similar to the standard GWAS setting. So this all sounds fairly simple and nice, but what are some of the challenges that's actually unique to these EWAS studies that's not in the genome-wide association studies? So one of the most significant challenges for epigenome-wide association study is this problem of cell type heterogeneity. Right? So in these studies, the samples we collect from individuals are typically very heterogeneous mixture of different cell types. Right? So for example, for the arthritis studies I'll come back to, we collect blood samples from these different individuals, which is made up of whole blood, which is made up of many types of cells. So for concreteness, here we have an illustration where we have just two types of cells. Yellow cells and black cells collected from each sample. And this can cause problem if there's a systematic shift, right? There's a systematic bias in terms of the composition of the cell types between the cases and controls. So in this example, the cases here, the patients actually have a lot more yellow cells, whereas healthy controls have a lot more black cells. And you can ask, okay, so why does that happen? So that actually happens in many settings, especially if the phenotype that you're studying, let's say, for example, is something related to autoimmune disease, and the sample you're collecting is blood, right? So the phenotype itself directly shifts the cell type composition of your sample collection. So that's actually quite a common problem that we see. So notice that, this is not such a big problem. We're just doing the standard GWAS setting, right? Because the genotypes doesn't depend on the cell type composition. But as we've seen from Jason's talk and from other people's talks, that the epigenome actually can be quite variable, and this is a problem that's unique for these epigenome-wide association studies. So just to define the problem very precisely, I want to go through what exactly we're looking for here in these EWAS studies, right? So to do that, I want to define two types of markers: the disease markers and cell type markers. So by marker here, it's meaning a specific CPG or specific locus that we're interested in. So what is the ideal study that you want to do? Ideally, you want to take an individual cell of each type, right? An individual yellow cell from a patient, an individual yellow cell from a healthy individual, and compare the epigenome across those two cells. And if we just do the thought experiment, so ideally you scan across the genome, across most of CPGs, they'll be fairly similar. And there'll be some CPGs where they'll be differentially methylated in the yellow cell from the patient compared to the yellow cell from the control. Right? So that's what we call a disease marker, and that's what we're interested in finding, because they actually reveal something about the underlying biology driving the disease. So the disease marker can also be cell type specific, of course. So this is specific for the yellow cells, but not in the black cells, right? So there's no difference there between the case and control. So I want to contrast that with a different type of marker, which is what we call a cell type marker. So a cell type marker is something that does not depend on the disease status. It doesn't care whether it comes from a patient or from a healthy individual. It only cares which cell type you come from, right? The only difference is between the yellow cells and black cells. Cell type markers are quite prevalent. We'll see how prevalent they are for the CPGs, but the point is that they're not the ones that we're so interested in, right? Because the goal of the EWAS analysis is that we really want to discover the true disease markers without being overwhelmed and confounded by all these cell type markers. So that's what we're trying to do here. Of course, the data we actually collect is not in the ideal light setting. So we collect data typically in these bulk samples. And the observations we have are measurements of CPGs in these bulk samples, right? So if we just look at the observations here in these two different loci, they both look to be actually different between the case and controls. They both look to be differentially methylated. But they're actually differentially methylated for very different reasons, right? So the first CPG is actually a true disease marker because it is actually truly different between the methylation value in a patient yellow cell versus methylation value in the healthy yellow cell. Whereas the second one is actually just a confounded cell type marker. There's no difference between the disease status. The only difference is between the cell type composition. It happens that this individual actually has a lot more of the yellow cells, so that's why this marker looks higher. So more formally, one way to model this is to think that, okay, so the observation matrix we have this big matrix of methylation values that's collected, right? So each row of this matrix corresponds to a sample, to an individual in the study, and each column corresponds to a particular CPG in the genome. So it's a very big matrix. So the actual matrix we have can be modeled as what we call a latent variable mixture model. So the idea there is that it can actually be modeled as the product of two unknown matrices, right? So one unknown latent matrix corresponding to the unknown cell type compositions of each sample, right? So that's what this matrix here. And the other matrix corresponds to also the unobserved true methylation values in the individual purified cell type. Right? So this is all that we observe, and we don't see enough of this. So what can we do? Right? So before I go into the actual the details of the algorithm, I just want to give you some high-level intuition for what's going on here. All right, so there's basically two pieces of intuition. That's very simple. The first piece that, okay, so remember the goal is that we want to find the disease markers and avoid all the cell type markers. So the first piece of intuition is that, okay, there are going to be many cell type markers. And the second piece is that they're going to be highly correlated with each other. So we don't know what they are, but there tends to be very highly correlated. And the idea there is that if there's a particular type of cell that becomes more abundant, all that all the markers, all CPGs that's specific for that cell type are going to go up together. And if that cell becomes depleted, then all those markers will come down together, right? So they're naturally correlated with each other. So here's the geometric idea of what we're trying to do. So Y is this vector of the phenotypes. So in this case, this binary vector of the disease status. So it's an n-dimensional vector where n is the number of patients or number of individuals that we have. So each of these x's are one of the measurements of the particular different CPGs, right? So it's also measurements across all the individuals. And if we have the CPG values measured at these cell type specific CPGs, then they tend to form these highly correlated cones in this high dimensional space, right? So they form this very relatively low dimensional but tightly correlated cones. So if a new marker comes along and we want to test whether this is truly associated with the disease phenotype, if it happens that this marker lies close to this low dimensional cone, this cone of cell type markers, then we know it's likely to be not truly associated with disease. But if it's actually correlated with the phenotype in a way that's orthogonal to this low dimensional cone of cell types, then we know it's much more likely to be a true disease marker. Right, so that's the basic idea of what we're trying to do. So more quantitatively, so that seems like a reasonable thing to do if we actually know what these cones are, or if you know what these cones of cell type markers are. Of course, we don't observe that. But the idea is that because they're so highly correlated, we can implicitly capture them based on looking at the correlation matrix among these different markers. Right, so basically if we take a look at the inner product or the kernel matrix of all these different CPG values, one thing that's very interesting is that you actually notice that there are these quite strong blocks of correlation structure along the diagonal of this matrix. Right, so these blocks are exactly corresponding to these geometric cones that we're trying to, that correspond to different cell types, cell type markers. Okay, so that's the basic geometric intuition. So how do we actually do this in practice? So the typical associations analysis is basically run these regressions of this form, where the Y here is again the vector of the phenotypes. The X here corresponds to the methylation value at a particular CPG, a particular locus. And the beta here corresponds to the effect size we're trying to measure. Right, so this is the basic association setup. So if we actually have all the true cell type compositions, then ideally the analysis that you want to run is to simply include all these cell type compositions as an additional matrix that we call C here of the cell type composition matrix, right? And then just rerun the analysis correcting for the cell type compositions. So that's the ideal scenario. But of course, the challenge is that we actually don't know what the C matrix is. So how do we approximate that operationally? All right, so this ends up being equivalent. You can write this also in terms of thinking of the phenotypes being sampled from this multivariate Gaussian. And the technique that we're going to use here is, I guess it's in machine learning literature, it's called Gaussian process, or it's called linear mixed models and statistics. But the basic idea here is that we're just going to approximate this unknown matrix of cell type composition by precisely what we saw earlier, right? This kernel matrix that captures the cone-like structures among these different markers, right? So we're just going to approximate it, and this can be computed very efficiently just from looking at the inner products of the observed biomarkers. So the parameters to be estimated here are the effect size beta as well as these two variance parameters that comes from this model. So this is since this is how the algorithm works, it turns out that mathematically you can show that this algorithm actually does the right thing under quite general situations whereby by that thing it's actually technically supposed to capture the cell type composition. But of course we have to see how does that work in practice, right? So as the exploration and validation of data sets, so we look at a particular study with our collaborators on rheumatoid arthritis. So it's an autoimmune disease. So in this study, our collaborators collected whole blood samples from about 350 cases and up to 300 controls. So for each sample, the DNA methylation value was measured across 450K CPGs. right? So what's nice about this particular study as a validation for us is that here we actually have the true cell type composition from each individual. So that can be measured from cell sorting and from other experiments. So we actually have this ground truth, or at least as much as possible. So here's basically how the algorithm works. Basically, since we know the ground truth cell type composition, we can actually run the ideal association study, or we use the cell type composition as additional covariates. So basically, that means that for each of the markers, so each CPG corresponds to a dot here. So for each marker, we can actually generate the ground truth sort of log p-values from this association study. So that's the x-axis. The y-axis corresponds to the estimated significance or estimated p-values using our method, which does not know any of the cell type composition. And the point here is that we want to see how well they correlate with each other. So in this study, it turns out that there are actually 5 genome-wide significant CPGs, genome-wide significant loci that we found. That's basically changes in CPG that are associated with the phenotype. So it corresponds to 5 red dots here. And they basically are exactly the same 5 dots, the 5 CPGs that are found in the ground truth study, right? So, and we also have very similar p-values. And in contrast, here's what happens if you actually do not do this correction. So if you just run the standard association study, then what you see is that you get all these very highly significant loci that completely overwhelms the true signal. But all of these guys are basically cell type markers, right? They're all confounding. So other way that people like to look at this is in terms of these QQ plots, where the red line here corresponds to what you expect to see when there's actually no true signal, when there's no true association. Right? So you expect most of the CPGs to not be truly associated with your phenotypes, arthritis. So most of them actually do, in fact, fall along the red line. And any deviation that's above the red line corresponds to truly significant markers. So this is, again, what happens if you run the ground truth, the QQ plot. This is what happens if you run our E-washer method. And this is what you get when you actually do the uncorrected study, where you have these hugely inflated p-values. So what it means is that almost every marker that you test in genome looks to be associated with a phenotype. But of course, that's not true. Just what happens just because there are these cell-type composition differences. So I should wrap up. So we applied the method to rheumatoid arthritis to discover these different biomarkers. And these are also being biologically validated. We also applied it to various other data sets. And there's a lot of ongoing work in trying to do this, basically extend this methodology and apply it into all sorts of other settings I'll be happy to talk about offline. And the algorithm is actually quite efficient. It basically runs on the order of a few minutes for each of the data sets on like a standard laptop. And lastly, just to summarize, so I hope to show here that when you do these epigenome-wide association studies, that to correct for cell type heterogeneity is really an essential part of the analysis process, right? So that's not something that you think about for GWAS, but it's very crucial here. And we present a method that automatically tries to correct for these cell type heterogeneity. We looked at different applications and validations in terms of DNA methylation studies. So if you're interested in reading more about this, so most of the material from the talk comes from this paper, and I'll be happy to answer any questions. Thank you.
Questions for James? Over here.
Thank you very much for a very interesting talk. I was going to pose a question. Even if you do correct for cell composition effects, in a given study, methylation in one CPG could be going up in one cell type, could be going down in another cell type. So a heterogeneous sample might not be capturing individual cell type effects in these. I wonder if this makes, if there's any way to account for this.
Right. Yeah. So of course there are other potential confounding factors. I guess what the point that we're trying to make here is that if you do not correct for the cell type heterogeneity, you're likely to be so overwhelmed with all these false positives that there's no hope of even trying to find what are the true signal. Right. So the idea is, okay, so after you first get rid of this bulk of the false positive confoundings, then you can zoom in on the true signals, and you're right in that there are all these other biologically interesting effects that will be interesting to model here. But first, we just have to really clean up the data analysis.
There's another and last question before the break.
Hi. I was wondering if you tried using it with sequencing-based methylation assays as well, where you might have like missing data points and coverage issues as well.
Yeah, yes, that's a good question. So, yeah, so we are also using it in combination with RBS sequencing data, and there are some interesting additional challenges in terms of, okay, so now you have instead of you have to sort of identify where are the signals and try to define the right windows to combine the data. So that's something that we're actually extending the method for.
What sort of end do you need to have a reasonable shot at using this?
So for the arthritis study, for the rheumatoid arthritis, the ends are on the orders of several hundred cases versus controls, and there we were able to actually find quite some of the more robust signals. And for some of the larger studies looking at, I mean, it's still smaller than the GWAS study, so it's on the orders of thousands of individuals.
Thank you.
Thanks.